Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Okay, we'll get started. It's an honor today to have Marty Tenenbaum join us. Marty is currently, he founded and runs Cancer Commons. It's a nonprofit open science initiative where cancer patients are treated, aimed at getting cancer patients the best treatments in accord with the latest knowledge, and with that knowledge, for the vision is continually updated on each patient's response, and you can find out more about that org. We'll be hearing about it, of course, at cancercommons.org. That's one word. Marty is a well-known AI researcher, internet entrepreneur. Beyond Cancer Commons, he founded CollabRx, based on getting the best treatments to cancer patients. He's a well-known internet commerce pioneer. He co-founded Enterprise Integration Technologies, which was acquired by Verifone, CommerceNet, Veo Systems, which was acquired by Commerce One. Actually, even before the internet craze that took over a couple of years later, Webify Solutions, acquired by IBM. He's served as an officer and director of MedStory, which our company acquired a few years ago. The MedStory, we had our healthcare solutions group, and he's a fellow and former board member of the AAAI. He was a consulting professor at Stanford in computer science for a number of years, and currently on the boards of Efficient Finance, Patients Like Me, and the Public Library of Science Plus, which we all know. He did his bachelor's, his master's, and his PhD. I should say, clarify, bachelor's and master's at MIT, PhD at Stanford. I don't want to get that wrong. People have different alumni in the audience here. Thanks a lot, Marty, for being here.
It's a great pleasure to be at Microsoft Research, and I have many, many friends here. I have some colleagues here. I'm here to try to enlist your help in the battle against this horrific disease. And what I mean by beating your cancer is we're really on the cusp of a new era where we can try to match precision-targeted therapies to the precise targets of a particular cancer patient based on information about what works best for people with those combination of targets. And to do that kind of, to get that kind of information, we're going to have to learn in a much more efficient way than has been traditionally possible through clinical trials or the clinical practice of medicine. And so informatics plays a large part, and the genomics revolution plays a large part, and it's the combination of those two together that we'll be talking about today. The paper is, the talk is in a real sense an update of a talk and a paper which I gave the Engelmore Memorial Lecture at AAAI in 2010, which became an article in AI Magazine in 2011. And two things have happened since the time this was written, both of which reflect growing humility when dealing with cancer. And everyone who deals with cancer goes through this transformation. First of all, notice the title, a computational disease that AI can cure. And what we're now hoping to do is be able to at least help individuals manage this disease through an evolving cocktail of drugs, much in the same way that AIDS patients manage their disease, but in a very information-rich way. And the second is that I no longer believe that this can be done automatically through AI. We're going to add collective intelligence as well. And so it's that combination that in fact makes the problem a lot more interesting actually to work on, and certainly a lot more realistically that we can get to places that can actually help patients. Let's begin at the beginning as to what this means to a patient who has just been diagnosed with cancer. Because literally every day I get calls from desperate, panicked people. My own sister on Monday called me up and said I was just diagnosed with breast cancer. And the words she used are just, just, I can't do better than she said, I'm numb, I'm paralyzed, I'm frightened. And this is, what happens in a cancer diagnosis is like you're fine. You walk into the doctor's office. He said he walks in. And in my case, he said, "Well, that was no normal node." The pathology report shows, and all of a sudden, you're not hearing anything anymore. You're thinking, "I have cancer." In my case, the doctor never used the term. Never said I had cancer. It wasn't a normal node. But suddenly, your life will never be the same. And at the beginning, when you're facing this horrendous learning curve, and you're just trying to educate yourself, and your doctor's telling you to do one thing, and you say, "Well, I need a second opinion." You go get a second opinion. They tell you something different, or a third opinion, fourth opinion. In my case, it was like a half dozen opinions, and they were all different. And there was no data on which to help make that decision in a rational way, and no way to recognize that you've at least considered all the possibilities. Such is the state of medical knowledge today. So patient is lost in this forest. And it's quite understandable if you think about how much information there is. There's a couple of million cancer papers, at least 100,000 published each year, tens of thousands of ongoing clinical trials. At ASCO, which is the American Society of Clinical Oncology, every year they have a national convention. It's held in Chicago at McCormick Place. It's one of the 2 venues in the world that can fit 30 or 40,000 people. They come in for a long weekend and they get a report on maybe 5,000 clinical trials, what the results were, right? And this goes on in like 20 parallel sessions all weekend long. And if you're not in the room at the moment that that particular thing was announced that was maybe going to be helpful for your patient, it might as well not have ever existed. You'll hear about it through the grapevine 5 years later. So the entire way that medical knowledge is communicated and disseminated, it can take literally up to a decade to diffuse from the major cancer centers out through the rural parts of the US, and another decade to get to the rest of the world. And this matters. What this slide is showing is that the 5-year outcome for major cancers, 5-year survival rate, can vary by up to 50% or more, depending on where you're treated and who treats you. Now, this is outrageous in the internet age. I don't know any other word for it, because it's largely about information arbitrage. It's like some people know things and other people don't know. And this is something that we really can and need to do something about. Yes, question?
How do you know the difference in survival rate here is causal due to the diffusion of information, not due to, let's say, high variance in the expertise of these different hospitals or talent?
There's certainly some procedures that that have expertise playing a big role: surgery, radiation oncology. But these numbers actually are largely about medical oncology, where we're talking about just administering drugs through infusion centers. And the difference comes from there, plus mixing it up with these other things. So yes, it's confounded, but the interesting thing is the more difficult cases go to the comprehensive cancer centers, and they have the higher results. So, there's one simple lesson you could say: if you get cancer, don't go to the Seattle Cancer Care Alliance or the Hutch, as opposed to some, not universally true, and I'll give you some counterexamples to it. But the big lesson is to take away from this slide: A, it matters, and B, we can make a really big difference. We can save a lot of lives with no new cancer science, just through better information. That, I think, is the real important message that I would take away from this slide. So I started Cancer Commons to do something about this, basically to provide cancer patients and their physicians and their families and caregivers with the information that they need to make better decisions and to get better outcomes, and to rest assured that they left no stone unturned. That really is the thing that really, causes angst in a family. Was there something else I could have considered that I just didn't know about? And we set it up as a nonprofit. I actually tried to do this as a for-profit first, and no one was willing to share information. It's hard enough as a nonprofit. This was born of personal necessity. I mentioned kind of in passing, 15 years ago, I was diagnosed with a metastatic melanoma, metastatic to liver. 15 years ago, there were no treatments that were effective, and the odds of my being here are literally 1 in 1,000. I should not be here. And I went to half a dozen doctors, local doctors, and everyone told me my odds were get your affairs in order. That was my odds. But they all told me something different that I might try if I wanted to try something. And so what's the data? And there was no data. And I was fortunate that at the time I was consulting for Rick Klausner, who was head of the National Cancer Institute, and he put me in touch with some of the amazing, clinical researchers that NCI had, both internally and they were sponsoring externally. And I talked to another half dozen or so people. If I had followed any of the advice of the local oncologists, I would be dead. At the next level up, the options were a lot more interesting. There were innovative immunotherapies, the beginnings of targeted therapies, and just the level of practitioners, surgeons who would be willing to undertake intrepid surgeries to remove cancers when everyone else would say it's too late, it's metastasized, right? But I can tell you one thing, formaldehyde kills cancer. If you can get it out, take it out, then worry about what else you can do. In any case, this was me, 12 months expectancy and no drugs available, and looking at a 15-year pipeline to get a new drug. And looking at all these different things, I said, if I get better, I can do something about that. And so this was going through all my medical guesswork. In the end, I just, made, on a hunch, bet my life on a clinical trial, and that clinical trial failed. But the trial helped a few people, and I was fortunate enough to be among them. And the question is, why me? I still don't know the answer to that, and 15 years ago, it wasn't possible to determine. But it is possible to determine now. Not that it would make any difference, because they took the vaccine that failed and they destroyed it. The lawyers made the company do it for getting rid of contingent liabilities. But the lesson was certainly ingrained on me that this was one more reason that once I got my health back, I had a mission. And the reason I would get my health back was to fulfill this mission, and that was Cancer Commons. If we fast forward 15 years later, and indeed just on the timeline that I showed you, on schedule almost in 2011, melanoma, simply because it's a relatively easy cancer to understand molecularly. It's a wicked cancer, but it's relatively straightforward molecularly, and it didn't have any good alternatives, was a place of choice for researchers who were trying to develop these targeted therapies that would hit particular targets, not try to just hit rapidly dividing cells like chemotherapies, but be able to bind to particular signaling pathways and shut them down. And so 2 amazing drugs were licensed in this 1 year, and there's been 2 or 3 more since then. So melanoma remains a hotbed. But now it's not the case that there's nothing that can be done. But that said, these drugs are hardly panaceas. They each took about $1 billion to develop. They cost about $150,000 a year. They're not curative. They're maintenance drugs. And almost all patients eventually recur, because what you're doing is the same thing you do in a laboratory if you're trying to sensitize a cell line to be resistant to a drug. You keep feeding it that drug, and the cells that survive, you feed it more of the drug, and then the cells that survive that become ultimately very resistant just through a process of evolution. So this really leads to the conclusion that the future, at least of this line of cancer attack, is going to be cocktails of drugs in which you try to anticipate the escape routes and head them off ahead of time and basically managing this evolving disease in the same way that one would manage an evolving disease like AIDS through an evolving cocktail. Try to stay one step ahead of the disease. Now, this, of course, puts the need for information higher than it's ever been. Information on what drugs should be applied to what patient. Clinical trials, which were long the gold standard for getting this information, are quickly becoming impractical in this world. And the reason is simply that we now realize that at the molecular level, cancer is thousands of distinct molecular subtypes. A single patient may have multiple subtypes going on even within the same tumor, much less at multiple metastatic sites. And if these need to be treated with combinations of targeted therapies, and the good news here is that there are hundreds of these targeted therapies coming down the pipe now. There's probably a couple of dozen that have been approved, maybe 100 more that are in trials of various stages, and a few hundred beyond that that are close to being ready to go into trials. But if these drugs need to be used in combination, then just think about the combinatorics. Thousands of rare diseases, hundreds of drugs that need to be used in combination to treat each of those diseases in a unique way. There's no way there's enough patients to populate those trials. We're going to have to find a much more efficient way to make use of what we learn from each and every patient in order to be able to populate this. And for that, I turn to what happens every day in community oncology, where millions of patients get seen by thousands of oncologists, and these guys experiment off-label all the time with drugs and cocktails. They don't work in universities, they're not running formal trials, they're not spending their time writing papers or IRB approvals or FDA, whatever. They're just doing what doctors do, which once a drug is approved, they can use it off-label, or if the drug's not approved, they can apply for a compassionate use to use the drug on a single patient, which is often much easier and faster to get than trying to get a trial approved, which puts thousands of people at risk. But essentially, nothing is learned from these exercises. Most of the time it doesn't work, so no one writes a paper, and even when it does work, the journals nowadays look down at case studies, which used to be a mainstay of the medical literature. Nowadays, it's all about trials. We've been ingrained to do that. So come back when you have 100 more patients and can demonstrate a statistical result. But that kind of statistical learning isn't that helpful. If this trial says that the drug is good for 50% of the patients, and this other trial says the drug is good for 20%, this drug is good for 20%, which drug should you take? Depends on whether you're more similar to the responders to this drug or the responders to that drug. The statistics actually don't matter much at the level of an individual once we have the molecular information to guide us. So, inspired by the experiments that go on every day with a desire to capture it, the vision for Cancer Commons is to build a model of molecular subtypes of cancer, this model, and how to treat them. And that model can be derived in lots of ways, whether it's Watson reading the literature or consensus opinion of experts. People talk about evidence-based or eminence-based. But regardless, you build this model up, and you select a treatment from this model based on the data for treating the patient in front of you, analyze what happens, and if the patient responds, that's great, notch up a success. If the patient doesn't respond, try to go in deeply. This patient now becomes a subject of research. Try to understand what's happening and what we can learn from it, update the model, and then go round again. And whatever we do, learn as much as possible and disseminate it rapidly. Don't disseminate it through the literature and through conferences, which adds months to years, not to mention the diffusion after the fact. The key is to disseminate this rapidly using internet methods and to make sure that the people who need that knowledge find out about it when they need it, as opposed to having to go look through the library of medicine for it, PubMed and so forth. All right. Diving down one level and looking at what these models look like, This is a model, a simple model of molecular subtypes of melanoma. There's about 10 of them here. And I could, if there was interest, talk about how these subtypes correspond to mutations in one or more of the signaling pathways that keep these cells in equilibrium with each other. You know, this pathway controls growth. This one controls proliferation and arresting of proliferation. MAP kinase, PIK3 pathway. These are color-coded, so mutations with yellow on them are mutations in this pathway and blue and so forth. And the more interesting ones have mutations in multiple pathways. And for each of these, there are treatments and recommendations which either come from data or from data mining, data analysis, or from experts. Regardless, this is a baseline model. And what happens is that patients come in, they get tested, they get assigned to one or more of these subtypes, well, let's say to one of these subtypes, and some of those patients will respond to the treatment and some won't. In that case, the subtype has to get split into responders and non-responders. And so over time, these 10 has actually proliferated. We now have 10 to 20, a couple of dozen, I would say, 20, 24, something like that. But it's not likely that this is going to keep exploding forever, because some of the newer subtypes are subtypes we have seen before in other cancers. This subtype of melanoma looks a lot more like a subtype of breast cancer than it does other types of melanoma, and there's a drug for it. And so the logical way to be able to do rapid learning is to be able to say, hey, this drug worked on this combination of mutations in this other cancer. Let's try it here, because fundamentally cancer is a molecular disease. So that's the direction that we're trying to head in.
Question.
Yes?
So for those, just curious, like the state-of-the-art treatment for those combination subtypes, are they simply just take the combination of the drug for the individual pathway, or?
That would be the simplest thing you would try, and often that's the case when there's two. When you have more, you try to do deeper network analysis to try to understand where these networks come back together, and if there's a place where you can, you know, if there's a mutation here and here, for example, there are two drugs for doing that, or I might be able to block something down here that would be able. So this is the kind of experiments that get done. And when I talk in my abstract about using drugs as probes to understand what the drugs do and how the biology works, that's the sense in which we're talking about it. It's, there's a lot of experimentation that can be done, and you can do this experimentation in animals, and animals are just not very good models. If you get something that works in an animal, the odds of it working in a human being or actually getting a drug out is pretty close to 1 in 1,000, the same odds I had. It's a very long, you know, there's a lot of things that go wrong. But if you have something that you can test rationally on a person who is out of options, so there's no ethics involved, you're in a human model, and you're in a human model, you know, years, perhaps a decade earlier than you would otherwise get. So not only is this a way to be able to inform treatment in the short term, but more fundamentally, it's a way to transform the way drugs are developed. And clinical research is done by tightly integrating research and care around individual patients. And that's one of the things I'm hoping to use Cancer Commons as a platform for driving this kind of systemic change in the medical community. Yes, Eric?
So today, what percentage of melanoma patients newly diagnosed even get this subtyping done?
Today, I would say a very large percentage.
Large do.
Three years ago, none. It's happened in a very rapid way. And I have to say that rather than the medical literature driving that, it's been the patients who find out about these things on patient blogs and drive the dissemination like wildfire. The urgency of patients just so far exceeds, you know, what happens once the academics get their paper published, you know, they're done, they get their credit. But the patients, when they see something that works, you know, they can upend research programs by saying to their doc, you know, I want to be tested for this thing because I just saw this paper on this drug that if I have it, I may only have a 2% chance of having that mutation, but if I do, you know, I'm cured, so I want to be tested for that. The researcher who published the paper had in mind another 10 years of NIH funding, you know, without having to deal with patients. But that's the power of patients. So what we did is create what I call a rapid learning platform, which is a way for all of the stakeholders to interact with the knowledge, with the data, and with the resources that are needed to both get the data and act on the knowledge. And so the way this works at a 50,000-foot level is the patients and the doctors understand what the options are. And they can come in with another option too. That's how it learned. Just what we ask them to do is do whatever you're going to do and tell us how it works so that the community as a whole can discuss and debate and keep updating this model. And the model also serves to organize the services and information of all the different partners. So we're going to talk about this a little bit later in the talk, but I want you to think about running this loop 30,000 times a day and learning, and doing it not just in one cancer, but across all the cancers, so we get this cross-learning. So now I'm going to, that was like a 30,000-foot level. Let's go down and ask what happens when we're dealing with an individual patient who has not responded to standard therapy, and what is it that genomics and personalized medicine, as you heard it, can actually do for that patient today. I think at this point I'm going to spend just a minute talking about 3 generations of precision oncology. The first drugs I showed you, verafamib and ipilimumab for the melanoma, these drugs came out with companion diagnostics. If you had a certain type of BRAF mutation, you were eligible for the BRAF inhibitor. If you had a certain type of thing on your T cells, you were eligible for the immune blocker. And so these companion diagnostics I would call Precision Oncology 1.0. This is Precision Oncology 2.0, and it's just beginning to be rolled out at the leading cancer centers and a few independent labs like Foundation Medicine that are set up to do this. So what happens is the patient who is, let's say, out of options gets biopsied, and basically you take a specimen from the tumor and a specimen of normal tissue, in the case from nearby. So in the case of skin cancer, it'd be maybe a sample of skin. And then what you do is you run both of these specimens, you extract the DNA and you run them through next generation sequencing machines, which give you a read of, if not all of the sequencing, at least of the exomes which code for the proteins. Then what you do is you compare these 2 sequences to see where they differ. And those are the characteristics that are distinguishing the cancer cell from the normal cell. Some of those may be significant in driving the cancer. Those are called drivers. Others are known as passenger mutations, which are just manifest as a result of errors that the cancer causes, but they're not driving the cancer per se. So the goal is to identify the driver mutations and then do some research. And typically this is literature research in PubMed to find out which of these differences, potential drivers, are actionable in the sense that there are drugs that are available to target them, either approved drugs or investigational drugs available in a trial. And then if there's time, one can test these hypotheses in either cell models. You take cells from that patient or from a similar patient and you culture the cells, make a cell line, and you test whether or not these predicted drugs are knocking them out. Or you can implant those cells in an immune deficient mouse, and this is perhaps a more accurate model. And in the future, people are developing analytic models of how drugs interact with signaling pathways in order to be able to predict what might happen in that patient. This is a little bit early because these models, while they exist, haven't been thoroughly validated. And there are commercial services that will provide in vitro or in vivo testing. Once you decide which is the drug you want to do, you treat the patient, and then you monitor, not whether the patient lives or dies in 3 years, but whether that tumor is responding to therapy on like a 6-week or 2-month basis using a combination of biomarkers or imaging. Did the tumor physically shrink? Did it physically become less hot on a PET scan? Or did the numbers go down if you're actually tracking markers like that are correlated with the cancer levels of certain antibodies that are associated with the cancer. And if the patient starts to recur, then you have the opportunity to do some more science. What you do is you do another biopsy comparing the treated cancer cell with the original cancer cell. What changed since the patient was responding? And now you go sequence those, and you find the salvage drug. So this is Precision Oncology 2.0. And there's been some remarkable success stories in 2.0. This one is one of the most famous because it was written up in the New York Times. Lucas Wortman was a leukemia researcher. He looks older. Was 28 years old a few years ago when all this came down. And he contracted the disease he was researching, which made it a good subject for a newspaper article. And he was on his deathbed. Nothing was working. And so people in his lab decided to do the kind of precision oncology 2.0 that I just described, and discovered that the guy's cancer overexpressed FLT3, which is a particular kind of protein, which is relatively rare in ALL, but relatively common in kidney cancer. And there's a drug that actually is active against that called Sutent. And so they put this guy on Sutent, and literally within weeks, he was back in business. And I believe he remains in remission and on Sutent to this day. You don't always have such happy results. A very sad case involved a personal friend of mine who was one of the world's leading glioblastoma, brain cancer researchers, who contracted pancreatic cancer. He worked at a Swedish hospital. And last summer, those of us who were working with him in his own lab, his own research fellows, were told that there was a VIP patient, not that it was the master. They were doing deep analysis. They discovered a potentially actionable mutation. They actually tested, since they had a lab, they built avatars and tested the drugs. They weren't wiping out the tumor, but they were controlling it, slowing its growth, getting stable disease. And so there was this drug they needed. You can get these drugs for research purposes. Getting them in an approved form to put in patients is harder. You have to get them from the drug companies who make them. There were 4 companies that made PIK3-mTOR combination inhibitors, and 3 of them turned us down. One of them, Pfizer, came through, gave us the drug. We did get the FDA approval to put this drug in this patient within 48 hours, and it did stabilize the disease briefly, but he was already, unfortunately, too weak, and he passed away. But this is the front lines of this kind of research. The goal is for every patient to learn something about the drug and about the molecular models so that we can make this better and better for each new patient. And the knowledge that we accumulate, Gleevec is kind of the poster child of targeted therapies in the sense that it was probably the first one that really had a widespread major impact. It was developed by Brian Drucker at Oregon Health Sciences University. It attacked chronic myelogenous leukemia, and basically it took patients from their deathbeds again to dancing in the streets within a month. And someone discovered that this drug also bound to a different target for gastrointestinal stromal tumors, a target called c-KIT, which was a surface signaling protein, and it had the same effect there. And this is another place where patients spread the word by wildfire, completely upended the plans of these guys who were planning to slowly roll this stuff out. And I notice here that in 2009, this drug was approved for 1 or 2% of melanoma patients who have a KIT mutation. So these people have a fundamentally different disease than I had. I didn't have a KIT mutation. And so someone actually asked the obvious question, like, tell me about the melanomas these guys had. It turned out they were all patients who had acral melanomas, which are melanomas that form on the soles of the feet or under the nails, other places where the sun don't shine. So it's a fundamentally different disease. It involves melanocytes, but a different disease. And the fact that the same melanoma doctors were trying to treat this using melanoma treatments was like, someone said, why don't you try Gleevec and melanoma? And someone said, I already did. And Gleevec, and I already did, and it fell. It only helped 2 patients out of 50. So someone raised their hand and said, like, tell me about those patients, and then everything started. But the point I want to make is that the connection between KIT mutations driving a small subset of melanomas was published in a research paper in 1991. There was nothing you could do about it. It just was forgotten.
I was going to say that I believe that even for the aquamelanomas, that sun exposure is considered a risk factor, and it's a mysterious connection to immune, they think, even though it comes in places on the body that doesn't receive the sun.
There's all kinds of things like that. The big point to make here is that people knew in 1991 that KIT mutations were a factor in these 2% of melanomas. They knew there was no drug. CML was developed in the late 90s, approved in 2001 in record times, the fastest drug ever approved through its process, like 2 months. They discovered that it worked on c-KIT, but now it took most of a decade to apply it to melanoma, right? Machine inference could have connected those dots and should have connected those dots. And we should have, in fact, even back here, before this, when we saw what this drug was, and if someone had, computational chemistry expertise and could look at and predict that the receptors, that this drug could bind to the receptors of these other oncogenes, and we could hit this off. Anyway, subsequently, this was approved in one trial for 6 different types of sarcomas. It's just a very, very interesting drug. In the back.
So has this led to a redefinition of success for human trials? That if 1 person out of 1,000 is cured, that rather than being declared failure, there's a further investigation into what that mutation or effect was, and then is considered a success, because then there's some population of people that...
The answer is no, it hasn't changed. That clinical trial would be considered a failure. However, bright researchers, when they see things like that, will say this is an extraordinary opportunity to do research to find out what disease that person had that we cured and how many other people had that disease. And then a platform like Cancer Commons, with its ability to have lots of patients who have been profiled, and we can connect up those patients with this researcher to test that hypothesis, not in a clinical trial, but the next day. And sometimes those hypotheses can be tested even retrospectively on existing data. And I'll give you examples of both of those before this talk's over. So you're absolutely right. That's the way trials need to be done, because they're not really trials at all, or maybe a better way to say it is every patient is involved in a continuing trial. This is an example of negative learning. So this is the drug vemurafenib that it was the melanoma drug for BRAF, approved in 2011. And it attacked a very specific mutation, V600E, in this BRAF gene. Very specific. And then someone found that very specific mutation existed in some small subset of colorectal cancer. And so everyone got excited. Do we have suddenly a solution that can stop for a while that kind of colon cancer? This drug didn't work at all there, or in very few patients. And so clearly there's other things going on. BRAF 600E is not a driver there. It's been preempted by something else downstream or cross-stream from it. But even that negative learning is very valuable. Why? Because, for one thing, it will save thousands of other patients from, it looks like a good hypothesis, let's try it. And people will try it over and over again, keep failing over and over again. They're just chewing up patients as if there's so many data points. That's what happens in clinical trials. Just use up patients without using them wisely to learn as much as you can and doing as much as you can for that patient. And the second thing is that the company that developed this will now go off and spending a lot of money trying to set up clinical trials in colorectal cancer. They'll go put their resources into more productive things. So whether the knowledge is positive or negative, it's just very, very important to capture and spread. I want to take a small look into the future, into the near future, which is, let's call this Precision Oncology 3.0. This is going much deeper into understanding the biology than merely trying to identify mutations and comparing them with normal tissue. And I will explain what this is, and that 2020 is not that far off now, it's 6 years off. But this is going to be transformative, and I'll show you what the difference is. So instead of biopsying, a few cells from the original cancer, with this we recognize now the heterogeneity, the full heterogeneity of cancer. So we want to take a sampling of cells from from one tumor and all the other tumors, so we understand what we're working with. And then we do very high coverage sequencing so we can pull those apart and understand how many different mutations that we have to deal with all at once, understand which are the most dangerous ones, prioritize those. Sequencing will be replaced by panomics. So not just mutations, which are one start of the information that you can get, but gene expression, metabolomics, epigenomics, all of the different things, proteomics, all the different things that can be measured, should be measured. At this point, you're probably up to at least 1 terabyte of data per patient. So we can now do big data on one patient. Yes?
How does the heterogeneity occur?
That's strictly Darwinian evolution. One expects the tumors are cells that are quickly replicating. There's 3 billion base pairs every time that happens, and mistakes happen. And as cancer cells get more and more advanced, their ability to correct mistakes also gets less. So these mistakes accumulate. And so there's typically a tree in which mutations don't go away. Once they occur, they stay on the tree, and then new ones show up. You have follow-up?
But within a single tumor.
Within a single tumor.
So is the thought that as the treatment or even the body starts to perhaps defend against the cancer, then there's a response?
That's half of it, and the other half of it is just strictly mutations as DNA gets copied over and over and over again, 3 billion times.
A terabyte is about $50 to $100 today, not counting the cost of generating it. You have an estimate what it will cost in 2020?
They've been estimating. I think the price of the testing will dwarf the cost of the administration of getting the specimen and signing the paperwork and all that other stuff. And I think that getting this data will be not the problem. There will be a problem in getting people to share the data because people think it has proprietary value, and the big cancer centers are very reluctant to share. We'll talk about that in a second as well. So we do panomics. The comparison is no longer a simple comparison, but a deep network analysis. So this is a chance for the Bayesians in the audience to really say, I want to understand what's going on at all of the different levels of knowledge. If I have gene mutations, that's going to predict possibly some differences in expression. I need to see that they're there. I have to see that the proteins are there that are causing it. I've got to see that these things are lined up. And that's the deep depth of analysis that is done at a handful of places today. One of the leaders is a fellow who developed his skills at network analysis in Seattle, which is Eric Schadt, and went off and is now running a center at Mount Sinai Hospital in New York. So as a result of this analysis, you're going to find the need to be able to find multiple mutations that you're going to try to simultaneously target. You can think about this as playing chess against the cancer, right? I move here, and the cancer is going to come back here, so I'm going to block it there, and then it'll come back here. I'll try to preemptively block it there. And the one thing I can say is that computers are better than most people at playing chess, certainly better than most doctors. So this is a place where computers are going to have a major impact. Testing is, I predict, increasingly going to be done in silico, because you can run all kinds of experiments in the course of an afternoon versus the really problematic things of getting cell lines to grow and getting them to grow in time to be able to test things for a patient. So you vet the test. Treatment is going to involve cocktails, and then monitoring increasingly is going to involve biomarkers as we get smarter about seeing what these proteins, these tumors themselves, are throwing off into the bloodstream to do their nefarious work elsewhere in the body. And we'll be able to monitor progression and then, of course, combine. So one comment I want to make about this slide is that everything on this slide is available today. That's why I can make these predictions very accurately. They're just not available at one place. And so one of the things that a platform like Cancer Commons can do is connect the places which have, the best laboratory assays with the places that can do the best computation, with the places that can do the best analytic, avatars, silicon avatars to analyze, and so forth. And to be able to take the learning from each of these patients and be able to combine them so that we can immediately learn from a handful of patients what might be good for the next patient. And that allows things to, if they work, to be quickly replicated, and if they don't work, to be quickly killed off. And thereby to do literally 10 years of research in human models rather than mice. And ethically, because in cancer you're typically dealing with people who are beyond standard of care. That's a way to make a real difference for cancer patients. So I'm about to start the second half of my talk, which is going to talk about informatics. This was your kind of introduction to cancer and personalized oncology. So before I go to the second part, let's take more questions on this one. Yes?
So one of the questions that I had was, so instead of the actual DNA level, cancer cells actually develop various different mechanisms for becoming resistant to drugs.
Yes.
Autophagy is one example of it. Another one is they actually create channels where the drug gets pumped into them and gets pumped out.
Yeah.
So where does that fit in this?
It's it's a complication, and then the way it fits is there are drugs that one has that try to shut down those channels. A good example is that cancers try to hide themselves. They become stealth from the immune system, and they do it by sending out proteins that try to tell the immune system to shut yourself down. And why there are receptors on the immune system for that is otherwise, if you have, an autoimmune disease, you'll get, or if an infection, you'll eat up the infection, and then you'll keep going and eat up normal tissue. So at some point, signals have to come out and say stop. And cancers have figured out how to take over those kinds of signals. That's just another example of what you're talking about. And there's a class of drugs that people are developing, immune modulators, to be able to preclude those sorts of things. So it adds to the complication, but it's something that absolutely needs to get factored in. And not all drugs. Autophagy is where the cancer cell, eats itself in order to be able to get energy without having, once you kill off its blood supply or something like that. And not all cells are capable of that, and the mechanisms you use may be in a different channel that will shut it down from wanting to replicate in the first place. It doesn't become a question of energy anymore. Question in the back.
Sort of a social rather than scientific question. I mean, as an engineer, I look at this and say, great, and then I think about the bean counters in my insurance company. And wonder how all this custom work is going to get funded.
One of the things that we're trying to do in Cancer Commons is bring all of the different players to the table and get them to look at this, what I call systemic problems, in terms of, it's not just how you get paid, but it's the regulatory approval mechanisms. It's how you get access to the drug, because if I said, there's only one company out of 4 that was willing to give us a drug, and we were lucky there. There are nice solutions which align everyone's interest with the patient, which is the key thing. And I'll just summarize what I think the nicest solution is, and we're working with all of the different players to try to move them slowly in that direction. The nicest solution is, if there's strong scientific evidence that some drug works in a patient, and you even have proof in a laboratory model or a computer model, the drug company provides the drug for free. And if it works, the insurance company agrees to pay for it because it worked, so it should be reimbursed. And if it doesn't work, they don't pay for it because there's some implicit guarantee here. The good news is that the drug company doesn't lose very much. The value is in their IP, not in the powder. So if it doesn't work, they write off the cost of the powder, and it actually saves them a lot of money in terms of wasting more money on 10 more years of mouse studies to try this good idea which seems so promising. You try it, you get past it, and you move on. And I think the payers themselves are going to find the data from precision oncology very valuable because in reality, 70% or more cancer drugs just don't work for the patients that they're prescribed for. And we can do a much better job just at the pharmacogenomics level of being able to see how people metabolize drugs, much less the matching to targets. Yes?
It seems like these low-level, you still have to motivate them the producers to attempt to research in all these various areas. My personal background here is that I had a rare sort of brain tumor where, there are only 10 people a year on the continent who get this. And so what that means is in the 20 years that I've survived, no one has come up with any new treatments for that because there's no market.
Yeah. This kind of a platform will, it's not like the cost comes in inventing new drugs, but the good news is, at least for cancers, that the cancers in all different parts of the body, including the brain, keep reinventing the same pathways. So over time, there will be drugs that will likely be applicable to any particular rare tumor. And the question is to have a systematic way to be able to identify what those are for a particular patient in need. So if you were ever, God forbid, to have a recurrence, you could use these techniques very likely to find, existing drugs that might be helpful. And then it would be my goal to try to help you obtain access to those drugs for compassionate use, which would be very helpful not just to you, but to all patients who had similar disease, as well as to the drug companies who suddenly would get a new market. And when for rare diseases, they charge through the nose for this, and the insurance companies pay it. So, if it works, the insurance company should pay it. Question?
You used two terms, compassionate use and standard of care.
Yes.
Are you saying that, does a person have to run out the normal treatments before you can go to these?
Usually that's the way it's done, and that's where the lawyers do get into the act, because if you don't do standard of care, the doctor can be accused of not giving you the proper treatment. But one of the problems is that standard of care typically doesn't work very well, and it could wind up making you much weaker, and or taking your cancer to a more advanced stage, where investigational drugs really should be used earlier. That's once we build up our confidence, demonstrating, and we do have to demonstrate that this stuff really does work more often than it doesn't work in terms of giving patients better outcomes, then we can get our confidence up and start moving earlier. But I think, in my case in melanoma, I ignored standard of care completely because I realized 1 in 1,000, if I tried anything standard, I'd get standard results and I'd be dead. So I didn't even consider standard of care. I just jumped. And I think you'll see more and more patients who want to aggressively fight their disease take that same outlook. You. Then I have till 3 total. So
on the record, yeah.
Yeah, because I want to make sure we talk about informatics, but so 2 more questions, then we'll move on.
Okay. So why haven't the drug producers and insurance companies haven't come to this agreement that you just described by now?
On their own?
Yeah, on their own.
Because the whole system is set up to approve blockbuster drugs that hit a very large number of diseases, one at a time. And the FDA won't even allow these drugs to be used in combination until they get approved as independent agents, because they think it's too tricky. We got to make sure they're safe and they work, and then we'll combine them. The real problem is with AIDS drugs is that the individual drugs don't often, they may be safe and they may be biologically active, but they're not efficacious by themselves. The early protease inhibitors didn't cure AIDS and didn't control it. They slowed it down a little bit. And the AIDS activists got the FDA to license these things in this kind of premature condition, at least as far as the FDA was concerned. And that changed the game, because suddenly the physicians and the patients had this small pharmacopoeia, this toolkit of molecules that they could start mixing and matching, and out of those came the cocktails which were effective and which are now, the standard of care. And by the way, virtually all of these cocktails are designed for a particular HIV patient by computer. And it's all computerized now because no human physician can keep up with this stuff. One more question, then I'm going to go to the second part. Go.
So you mentioned like this clinical trial that have failed, but they contain sort of a small portion of successful case, and those, if you can go in and compare their molecular profile and whatever, then you might be able to figure out A, what are the biomarkers that distinguish them, and B, what would be the targeted promising. But how, like, as of today, like, do we have access to this kind of failed clinical trial data?
We're beginning to get access to failed clinical trial data. Not successful one, but most of them fail, so there's a lot of data. I think, let me, in the second part of the talk, I'm going to talk about what we're doing, which is really a big part of what I'm doing to try to figure out how to pull the data out of the institutions, kicking and screaming in many cases. So we're going to move to the second part of the talk, which is the informatics and how this is going to make a big difference in terms of applying these ideas at scale to be able to really help millions of patients. And in this paper, I'm going to summarize the conclusions of this paper with a set of grand challenges, which were at the end of the paper. It said the goal is to organize the world's knowledge of cancer and use that knowledge to basically design treatment plans for individual patients, and then to coordinate those plans so that in an ethical way, this is the exploration trade-off, in an ethical way to be able to do good for the patient but also good for science, right? And then to be able to integrate all the resulting data back across all patients in order to be able to very rapidly learn as a community. So think about what we're doing as organizing a vast adaptive search over the space of targets and trials and over the space of targets and drugs. And then using all kinds of knowledge and heuristics and pragmatics and ethics to be able to guide that search so that as a whole we can do the most good for the most patients, and always putting the patient first, by the way, and then, which is not what happens in clinical trials. Clinical trials about putting the drugs first, and then try to learn as much as we can to help other patients. So that's the goal, what we're trying to do, and I'm going to show you examples of how we're going about doing that. Starting with organizing the world's knowledge. So we first discovered, that's a lot of knowledge to organize. One approach to this is gotten a lot of publicity, which is IBM's Watson, who said, I'm simply going to go read it all. And they talk about reading 2 million articles of cancer. And there's some very good things about that, namely it's all automated and the knowledge is ostensibly encyclopedic. If there was some rare event, if some rare finding that happened to be relevant to some particular patient that was reported at the San Antonio Breast Conference that a few hundred people went to and most people didn't, Watson would have come up with it. The learning is continuous in the sense that they try to update the findings by reviewing the recommendations for each patient and how they worked. And based on, I should say, one person, Mark Chris, and that's one of the foibles right there, which is we want the collective understanding of everyone, not just the one genius that runs lung cancer at Memorial. And you can do inferences of the kind that I mentioned. So Watson, in principle, if they got its act together, could infer that Gleevec early on might have worked against the acral melanomas with the c-KIT mutations, the 2% of them. On the other hand, it might have even been able to do earlier if it could reason not just in terms of connecting the dots at the knowledge level, but also being able to go back and say, I understand computational chemistry, and I'm looking at the shape of the receptors, and I'm predicting this drug. You could imagine looking at all drugs. There's 5,000 drugs that have been approved, and there's something like 20,000 molecules that have been approved safe for use in humans that are not drugs. They failed for whatever reason or business reasons to not progress to be drugs, but they're still available. You could mix and match, so that's an opportunity. Okay, but there's also a downside, and I'll summarize the downside, which is most of the literature is either irrelevant, out of date, or wrong. We just don't know it's wrong yet. Next year we'll know it's wrong. So, and the fact is that the reason you don't see Watson, applied yet in practice is that it's still learning. I mean, the results that it comes up with are like a first-year medical student, and Mark Chris has to keep correcting them. It will get better. Watson's the way to go, or a way to go. But that's the challenge. Not having the resources of IBM, my colleague Jeff Schrager started working with a group called the Melanoma Molecular Map Project, which was an international consortium of about 100 melanoma researchers. And they did a kind of hand-curated version of this. Now, each of the lines in this spreadsheet represents a coding of the major result from a paper. And these papers were, carefully selected, curated by this group to say this was an important finding. And if you read this table in detail, it would say that the presence of some protein either enhanced or decreased the sensitivity to some drug, or increased the sensitivity to a second drug in conjunction with the first drug. This is kind of the level of conclusions of these papers. And so it went through, there's like 2,000 papers that they covered, and they included strength of evidence, which was very straightforwardly coded. Namely, if the evidence came from a petri dish, that would be 1. If it came from a mouse model, 2. If it came from a case report, 3. If it came from a small trial, 4, a large trial, 5, a meta-analysis of many trials, 6. You get the idea? And then the way they did this was if you had a patient and you studied what markers they had, you could decide what drugs you were thinking to be interested in, and based on that profile, you would then select the weighted evidence from the rows in this table, and you'd get a personalized drug rank. It was just that simple. A patient has certain, I have a drug, the drug patient has certain things. I'm going to look at all rows that talk about the patient's mutations and that drug, and I'm going to add up and the evidence down based on how strong the evidence was, and I will get a result. And this worked fairly well as an approximation to what Watson was trying to do, but it's hand-curated, it's not automated. We went further in Cancer Commons and in this company, Collabrix, that I did. We didn't take 100 people. We took one editor and a couple and a scientific researcher and a small editorial board of 3 or 4. And we basically hand-curated. We went to them and I said, how many subtypes of melanoma do you think there are? And how should they be treated? And then I went to a handful of other experts, and that consensus gave you a baseline model. But this was all this was intended to be. And then from this we could start to do learning, right? We can capture data as to whether it worked or didn't work, and start to refine these, split them. So there's various ways you can organize the world's knowledge, starting from encyclopedic to this, and obviously it'd be good to have some combination of them, because at the moment a handful of good doctors will have read all the literature and will have pre-compiled and digested that and make it available in this nice digested form. The one problem with this, again, and it's what I talked to Haifeng about, is that these are per cancer type. And I really want to do a pan-cancer model by just by molecular subtype, because that's the way we have to start thinking about cancer, not by organ. Talk about head and neck cancer. I don't know what the organ is. The reason that's a medical specialty is because the muscles and so forth require a certain kind of surgery when you're operating on it. So it's a subspecialty, but it's nonetheless treated as an oncology specialty as well. So you have to think about cancer at a molecular level. Okay, so that's knowledge. What do we do with this knowledge? On this platform, we turned this into basically an apps platform, and we developed a small set of apps to be able to do rapid learning. And by that I mean, for patients and doctors, the ability to look up what treatments and trials are recommended, number one. Number two, to be able to discuss those treatments and trials or treatment plan with peers. And number three is to report outcomes. And then for researchers to be able to capture those outcomes, analyze them, discuss those with your peers, discuss updates to the consensus model, and then to be able to update the model and rapidly recruit patients to validate results or to continue testing. And so this became a platform for being able to do this kind of basic rapid learning for precision oncology 1.0 and maybe 2.0. This is not the deeper stuff yet. We'll get to that in a second. But it's the thing that we need right now to be able to provide information to patients. So I'll give you an example of some of these apps. This is our first app, which was to capture information from patients. And this is just a wizard, basically, that captures information about, this is demographics. It talks about date of birth and postal code and blood type and things like that. The markers that you've had, the treatments that you've had, and the responses. And this is a timeline that shows you're getting better as you go higher if it's turning green. But you get a record of things. And as an incentive for patients to provide their data, what we had initially was an application which gave them targeted news and alerts to new studies that affected them. So, and in fact, this could be precisely targeted. And this is something I'd love to work with, researchers at Microsoft who actually care about wanting to do precisely targeted news. At the moment, we're matching keywords, and you see a marker and you get it. But patients are in a particular molecular subtype or a phenotype that's based not just on what their subtype is, but how they've been treated, how they've responded to drugs. Patients with the same molecular markers may respond differently to drugs. They're in a different phenotype. So if we could take the news and other information and classify it in the right way and then hang it there, so when a patient is asking for what's new for me, these are personalized dashboards, they get precisely the information that's right for them, for people who are in their particular state of the cancer journey. That would be very, very helpful.
It'd be interesting also to think about, even on these kinds of sites, you have some of these experts of the form that you went to that aren't available in the community come on and provide some information and some expertise.
Absolutely. To be able to put a social collective intelligence around this is absolutely the idea. And the reason I don't talk all that much about it is that we are committed to channel partners who do that for a living. That's all these advocacy groups. And my challenge is to be able to mine the patient blogs and discussion forums and QAs and so forth, go on all these sites, to pick the 1% of postings that are actually, scientifically important, and then organize those so that they show up so someone can find them. The problem is no one can find this stuff. And so if we can organize the world's social media, and this is something that I don't see myself having the expertise to do, I'd love to do it with you guys. That would be extremely helpful. This is concept pictures of where we'd like to go. As people report data, we have, in principle, the ability to be able to show people information they've never had before. Very straightforward information. What treatments are they taking and what the outcomes were. So, this is a simple depiction of I'm a patient and here's all the patients that are similar to me in some space. And instead of having a subtype, right, this is using nearest neighbor type analysis. And some of these people, you might not see it, but some of them are not smiling. These guys are smiling, so I think let's see what this guy did. And let's see what, for the patients like me, what treatments did they use, what the outcomes were. You can start displaying it in ways that really make clear for the first time for a patient who is just like you, has the markers, has been treated in the past with other drugs. At this moment in time, these are your options. And, more people, bigger circle, more people are using A than B, and D is the best because it's both the most efficacy and the highest safety. And so I want to go try that one. Similar, one can crowdsource other things. So you can imagine various kinds of apps like this. It's not my goal to write them all. It's my goal to create the platform and that people will use this patient data, send them to the apps, and the apps will come back with all kinds of interesting things. So now let's talk about where the data comes from. The patients will give us data if they have it, and they'll know, typically, a breast cancer patient will know she's triple negative or estrogen positive or whatever it is, but they won't know the detailed, and they can't know the detailed sequence information that comes back. So to do that, we've got to get the information out of the institutions that have that. And we're doing it in two ways. One is we're actually trying to sign up institutions to become network partners. And by that, there's a quid pro quo. We will send patients to partners that are willing to share data, and they'll send back the data for the patients who that we send them or that are part of this program. And what they get in return is something that's pretty important to them. De-identified data from all the patients in the program, identified data from their own patients. And why that's important is Foundation Medicine, for example, is a lab that does molecular testing. They give you recommendations. They have no relationship with the patients. So they have no idea whether patients have followed the recommendations or if they did, what the results are. So that's what we're doing there. And of course, they get referrals. So this is high value, and it's of high value, especially to the smaller institutions. MD Anderson may have enough data on its own to be able to do its own thing, although even they are beginning to recognize that on a global scale, they have to join forces with other people. Now, for some institutions, maybe because they're smaller and we don't have relationships with them, we have another vehicle, which is we use the patients as leverage points to be able to pry the data out of these institutions, whether or not they explicitly want to cooperate. And we have two things going for us, which we never had before. And for people involved in Health Vault, this is actually very important because there's two things that are happening this month that never happened before. One is that the HIPAA regulations have expanded to guarantee that patients can get access to their medical records, which they always could, but now they can get it in digital form within 96 hours. When I used HIPAA to get my medical records some years ago, they let me go in the records room with my folder, pick out a few things, and then pay 25 cents per to get it Xeroxed and a piece of paper.
Is digital written into the recommendations?
Pardon me?
Is the word digital
Yeah, it's in there.
written into the law?
But the problem is that it's likely to be, ASCII organized in some random way. So there's still good work to be done for people who want to take information and structure it, for sure. But the other thing is that on the flip side of this, the institutions have a real incentive to give them the data, because Meaningful Use 2, which is part of the Affordable Care Act, actually has positive incentives to a provider to have a third party, not themselves, but a third party make meaningful use of their data, and this qualifies. So the patients have a legal right to the data, and these guys have an incentive to give it to them because they get better Medicare and Medicaid reimbursement. So that's what we're counting on in order to be able to get data. And so once we have that data, then these same institutions, many of them are like commercial labs that might want to provide a second opinion app, for example, because they're MD Anderson, they want to recruit patients and show how great they are, right? And they'll get some patients out of that. Or clinical trial finders because you're a drug company and you have some studies that you want to do. I'm hoping that this will grow to be a very vibrant ecosystem of apps that are coming from all kinds of third parties. And my goal, again, is not to turn Cancer Commons into a site. I'm perfectly happy to white label all of this behind Health Vault or Bing or advocacy groups or what have you. The goal is to help cancer patients, not to build an institution to compete with them. This is an example of a third-party app that actually exists that came out of the company I started, which provides a little expert system that you can actually run it today and try it. It runs off of the lung cancer model that's similar to the melanoma model. You can try the melanoma one too. There's about 3 or 4 of these at collaborix.com.
These two advisors, how do they get involved?
No. These are the Fisher and Flaherty are the co-editors of the melanoma model that I showed you. They have a small editorial board, and they built the consensus model, and that's the way that came to happen.
But I'm curious why the head of Targeted Therapy Finder Advisors?
This is called Targeted Therapy Finder, and these are the advisors.
Oh, okay.
For it. We are trying to get a lung cancer pilot going, rapid learning, involving a number of institutions that are directly or indirectly associated with the University of Chicago and Oregon Health Sciences, which are the 2 charter institutions. And they have colleagues, and we're trying to just build a rapid learning pilot in lung cancer. But again, I want to generalize that quickly to all cancers, and so we're also trying to add institutions that by leveraging the Stand Up to Cancer, Prostate Cancer Dream Team, and the Melanoma Dream Team that I'm associated with, to try to build up the Cancer Commons network. And build a sustainable funding model for Cancer Commons by getting these people to all contribute a little bit to sustaining of the network, and then use this network to run their studies in a much faster way than they've been able to do before, and capturing the data from every study in order to make it available to patients. Okay, at this point, all of these things, planning the experiments, integrating the data, we're now moving beyond 1.0, 2.0 to really precision oncology 3.0, where we're trying to do deep reverse engineering of these tumors and integrating the results. And I just want to show you a few examples of how this works. Again, the trade-off between exploration and exploitation. You're always trying to do the best for the patient while at the same time maximizing others. You saw a picture of melanoma pathways. That was a severe simplification, and this too is a simplification, because this is just like a pool of thousands of proteins all interacting with each other in random ways. And I don't know how to depict it other than a cartoon. But the point is, it's too complicated for people to really anticipate all of the horizontal and vertical feedback among these pathways. So it really is something that machines should do. And this is what's involved typically when you're reverse engineering a tumor. You start with a genetic profile, which can include expression and other things. You, on the basis of that, make hypotheses of what proteins ought to be there. You try to line those up with pathways that are there, select targets and select drugs. And you're trying to come up with, if you like, a Bayesian inference across all these different levels of knowledge that are going to identify, based on the evidence that you have, what are the best targets, what are the best drugs I have for those based on the evidence. And there's various ways you can try to improve your ability to deconvolve all of this information. So basically you have multiple specimens which can help you put noise in its place. You're dealing with panomics, which allows you to do this multiple levels of hypothesis validation. The fact that probably the most useful level to start with is not mutations, but expression. And so you, based on expression, you look for proteins that you expect to be present or possibly phosphorylated. And then you can go back and say, are there mutations that maybe account for that? But in any case, you're looking for the master regulators. And the interesting thing, when I talk about, people talk about big data, and I'm talking about either small data or deep data, data from one patient. The point is that you have 20,000 genes, right, that you're getting expression data from. In a single patient, it's likely to be very noisy, any of those. But if those of you who are familiar with concepts like Huff transforms, any one of these genes can be noisy, but if its expression is over or underregulated, it'll implicate some number of problems here. The next one that's like that will indicate some number of problems, but they'll be scattered. And the real master regulator that's responsible will keep getting votes from all of them. That's the way this works, which is very straightforward.
So if you're getting full sequences and diffs, aren't you resolving lots of variables into beyond the truth, even with a little bit of noise on them, or is it not?
Yeah, no, the point is you will get a very strong signal. If you want to put a p-value on it, where the doctors in the clinical trials think a p-value of 0.05, 1 in 20 is fine. The p-values on dealing with 20,000 mutations to try to isolate where you think the action is like 0.0001. And
I was hoping that the sequence information would give you some direct knowledge.
Yeah, well, it's also the pathway information. You're trying to.
You have the regulatory stuff going on here.
You're trying to put it back in place in a way that makes a story that compares what you're seeing with what the public models are. So you may not, from a single patient, have the ability to be able to determine what is the de novo model of cancer. But you can look at all the public models and you can say, this is most similar to that model with the following deviation. And that's where big data comes in, as a kind of baseline that you use as part of this interpretation. Similarly, sequential biopsies. So every time a drug interacts, you want to figure out, why did this tumor not respond? Right? There's lots of potential reasons. Either the drug didn't reach the tumor, or if it did reach the tumor, it didn't bind to the receptor. If it did bind to the receptor didn't turn off the pathway as we thought it would, and so on and so forth. So this is, and response to therapy. So this is, using drugs as probes, as we talked about, in order to be able to infer in a deep way. And I'm not going to tell you guys how to do this kind of work. I just love your help in trying to work with individual patients with data to try to help those patients and in the process to learn. This is not something that should be done, in my opinion, as disembodied research, because the doctors are there working and they can use your help.
Has there been some good graphical models work of this kind of?
Absolutely. And Eric Schadt is...
with these N levels, for example, simultaneously?
Yes, and I can help you. I mean, I've been involved in this personally, and I can absolutely help you, but it's not something that I have the time or even the expertise to be able to do on my own. But we can certainly provide the expert help directly and through our own collaborators for people who want to work in this. And the second big one is, these are all the people doing big data, like the Cancer Genome Atlas, and this is ASCO trying to capture data from all of their oncologists. But this is this cumulative trial that I talked about, where we're trying to analyze and learn from every patient. This is my colleague Jeff Schrager's diagram, and I'll cut it down to size for you. This is what is happening when a patient is, a standard patient comes in, he gets tested for omics, and this standard treatment is available. They get treated and monitor response, and if not, you come back, you have more information, and you retest the tumor, and you go around this loop. But there are no options in the standard thing that actually integrate research. This is clinical. Now, if you take the first step of what we're doing, if in fact there are no standard treatments available, then you come down and you look at what are all the research options that are available. So in those subtype models, if there's 20 of them, if I sort them by evidence, the top 10 may be standard of care. The next 10 are like, hypotheses that someone wants to try. So are there some good things to try? And if there are, then you say, is this best choice for the patient? And if it's a good choice for the patient, then you go and treat. If there's no options at all, then you consider that a dead end. And if it's not a best choice, that is, the patient doesn't want it, then what we can do is go and say, the patient doesn't want it. It's not clearly one best choice. It's like two equal choices. Then we can choose the best one for science. So this is the priority that I was telling you about in exploration, exploitation. Namely, if it's a clear best choice for the patient, then you do it always. If it's not a clear choice, it's like there's two things and we have to have enough evidence to know which one's best. Everyone is doing this one, let's try the other one because we want to explore the rest of the field.
We have inpatient cancer patients who, given a set of targeted therapies, each with few side effects, just want the cocktail right away, and we'll see what happens.
You know what happens then? That's this guy, no options, right? You've seen this, right? This is where we give them. And now suddenly we have a whole new set of choices. And the goal of this analysis is to be able to integrate the evidence from every patient in an optimal way that gives each patient the best possible outcome for that patient based on science and drives the entire learning forward for the field. So I would love to build a global cumulative treatment analysis process on top of the Cancer Commons platform. We're not there yet. That's where I want to go. I'm going to skip this and come back to it if we have time after my 3 o'clock cutoff time, which is talking about what we can do beyond trials to change the way medicine is practiced. I want to spend the last bit of time talking about what we and Microsoft might do together. So Cancer Commons is building an infrastructure that will have data and knowledge and various network partners from pharmas to payers to providers, patients and whatever. We're bringing all these people together. And what we need are partners of many kinds. And I look at Microsoft as this extraordinary organization with, most importantly, 1 billion users, some of whom are going to be concerned about cancer at any moment in time. And you have some interesting properties, Bing and Health Vault, which are intriguing to me because Health Vault is something that can be turned into a registry for the kinds of data that we're collecting with our Donate Your Data. And Bing is, to the extent that it wants to become a decision support engine, can take that information, that profile, and be able to do true decision support using the kinds of apps that I was showing you. Those could be integrated. Similarly, if to the extent that Bing is actually going out and proactively looking for news for someone, this all fits. I want to build apps that can be used with Health Vault and Bing, and so the specific ask is I would like to do a collaboration with Health Vault, and I would like to get some Bing cycles or AdWords so I can start playing with that. With Microsoft, and this is for a company that on your health page explicitly says that the goal is better outcomes. We share that. That's what this is fundamentally about. As far as research is concerned, there are many, many things going on, and I've only listed a few of them in Microsoft research that are absolutely essential in order to be able to fulfill this vision. Hui Feng and I are working on, have been talking about building a pan-cancer knowledge base, largely mined from the literature, but also then corrected by human experts and ultimately by the data. Reverse engineering the tumors we talked about. The global cumulative trial analysis was that diagram that I just got through showing you. On the natural language front, there are two things that I've identified. One is patients have questions. They want questions answered about the information that we're giving them, and just putting up any kind of a display is not going to be enough. And some of these questions may need to be answered by machine, and some will need to be passed off to experts at Sloan Kettering or MD Anderson or the Hutch. And we want to be able to make that seamless. Applications that do crowdsourcing of second opinions is very interesting. You know, what do other patients who have been in your boat recommend doing, right? There's all kinds of, or physician referrals, not based on what, you know, your friend knows. I need a good doctor for this. But we have the numbers now. We will have the numbers that say for patients who are like you, right, who have been in with this molecular subtype and these treatments, these are doctors who have had the best outcomes, and these are the methods that they've used to do it. We'll have those numbers, and I want to be able to get them to patients. So with that, let me close on a personal note and say, for just one moment, forget that we're here at Microsoft Research or researchers at all. We're people. We're all going to be patients someday, or we're all going to be taking care of patients someday. It's just a question of getting old enough, and this is the scary thing about getting older. Suddenly you hit a certain age and all of your friends are dealing with this stuff. And when this happens, what you're going to want is to know that the collective knowledge of everyone in the world is available so that you will make the best possible decisions, get the best possible outcomes, and know that you've left no stone unturned. And so please, please, please join me in this urgent quest to kick cancer's ass. Thank you. Thank you very much.
Any other questions?
I would like, I know anyone who wants to leave officially over, but I want to go and show the one more slide if I can. And then it's this one slide. This is, if we can get to a point where we can get 100,000 patients on this, then we have patient power. We have an army, right? And what can we do with that army? Someone asked before, what happens if you have one responder in a trial? Does the trial succeed or fail? And the answer is, today it fails. But this is an example of someone who did it right. There was a trial of a drug, everolimus, for, I believe it was kidney cancer, that was ostensibly targeting mTOR, which is a well-known cancer target. And there's only one lady who responded, and they did the complete sequence of her tumor to find out what it was that this drug was hitting. And they discovered it was something downstream of mTOR called TSC1 that she had, and few of the other patients had. And so they then went back and looked at the rest of the data, and they saw that this TSC1 was present in all of the other patients who had at least a small partial response to, but a lesser extent than she had it. And it turns out there's lots of other cancers in which TSC1 is quite prevalent. So suddenly there's an opportunity to take a drug which otherwise might have been considered a failure and to go look at all the other, few percent of every cancer, and suddenly this becomes a major success.
Yeah.
So this is one example, exceptional responders. N of 1 studies. This is a true story. There was an investigator at Columbia University who was presenting at ASCO last summer in front of 2,000 people in a big hall. And he said, based on molecular results and reverse engineering and finding the master regulators, I knew exactly where there was an opportunity to interdict this tumor. And I got a hold of the drugs and I combined them and just knocking it out of the park, killing the tumors. Mice are doing great. And now I'm going to start a clinical trial. My plan is in 2014 to do the paperwork, get the IRB approvals, the FDA approvals, the drug company approval so I get access to the drug, apply for funding, at some point start accruing patients. Eventually you have enough patients that you can begin the study, and then you run the study for a few years, and then you write it up, and then you submit it to ASCO. And then ASCO escrows the results for 3 to 6 months because, God forbid, they should be presented before the conference. You're talking about a decade. I went up after the talk and I went to the microphones and I asked him, I said, so you're doing good with these tumors, where'd you get them from? He said, well, I get them from a tumor bank there. I said, whose tumors were they? He said, I don't know, they're de-identified. And right at that point, right, I raised the obvious suggestion that said, well, these tumors came from someone. If we could find out who they came from, you could, tomorrow, these guys are dying, so it's not a lot of, you can do one of these compassionate use things, and we can actually test your theory and decide if it's good or no good. And if it's good, we can replicate it on 4 more patients and then apply for fast-track FDA approval. And if it's no good, then you can go off and do something else and quit wasting the next 5 years of your life doing that. That's the kind of revolution that we can do with a platform like this. Final example, which is not unrelated, is that this is a drug, enzalutamide, which is used in prostate cancer, recurrent prostate cancer, to block androgens. And it stops the growth for a while, but these patients all recur because the cancer will find other hormones to drive it. And typically, often, it uses metabolic hormones from the metabolic pathways, such as insulin. And so you find metformin, which is a commonly taken drug. They've done some experiments to suggest in vitro, in vivo, in vitro, that these will, in fact, by blocking the insulin, make some of the tumors responsive again to enzalutamide. Now, of course, the answer is we've got to start a clinical trial to test that. And in this case, I had a similar response. I went up and I said, certainly there are large numbers of patients who are older men who are on this drug for their prostate cancer. Some have recurred, some haven't. Some are on metformin because they're also battling diabetes or not. Why can't we, in an evening, go back and crunch the data and get the answer to your question? So another example of retrospective studies that could be done very, very quickly on many promising things. The question is, there's so many hypotheses that need to be tested, and so few patients, especially as you get these subtypes of cancer. We have to find a better way to exploit the patients for the benefit of the patients and the benefit of science. And tightly integrating research and care is kind of what the end game is, with the aligned incentives that we talked about, so that the insurance company pays when it works and doesn't pay when it doesn't work. That's where we're headed. And the final little build here is that there are obvious examples for informatics to drive this. Namely, what we need to do certainly is to be able to look at all the results to identify exceptional responders from all the individual patients who are reporting one thing or another. It's not that they're all equally interesting. I want the ones who are going to teach us something, and then we'll really drill down on them. Here, there's an opportunity to be able to directly match patients and the researchers who are interested in them. And here we have an opportunity to build an e-trials database by integrating information from all the partners that Microsoft can get a lot easier than I can, so that we can do these retrospective studies overnight. So with that, I will again say thank you very, very much, and I hope we can find some very productive ways to work together. Thank you.
I was hoping to hear punchlines on Bill 2 and 3, like even though it's 2 months out from from ASCO meeting. I was hoping that this guy went back and figured out a way to go to the company and
You know, I will go ask him if I hadn't intimidated him enough. But there is an interesting punchline on this one, which is interesting because there was a different person who came up with the same result in a different institution. And they didn't know about the fact that this, I couldn't talk about it. It was a perfect example, but I couldn't talk about it because they hadn't published this finding yet. So it was like another 6 months till they publish it before they then start thinking about their trial. And then like the next week I was at the ASCO meeting. I literally went on an airplane from this meeting to the ASCO meeting, and I hear about another group that actually had a trial in process or in the process of starting up. And I didn't want either of them to do a trial, but nonetheless this one group was like, you can see how all of the replication and redundancy of medical research, the same experiments get done over and over again, and the people who lose are the patients every time because you only have one life to live. Why would you want to be in something that's known not to work?
So if today we want to do this kind of trial, e-trial, what's the bottleneck?
This one? Yeah. You know, I don't think, there's certainly no technical bottleneck. The term e-trials in precisely this way was actually coined by Andy Grove, who wrote a wonderful editorial in Science. And you can just Google Science and Andy Grove. And he caught so much flack for this. His article was saying, you know, the drug industry needs to be more like the semiconductor industry. We need more knowledge turns. And instead of, if something fails, you throw it out. If something fails, you try to see what part of it might have worked and learn from that and build. And his best idea was to say, I want to do e-trials. And so in order to do this, you really need to get access to a lot of data. But you might be able to do it by partnering with one organization. And a big pharmacy benefits manager like CVS Caremark will have data on 50 million patients. That's more than enough to answer a lot of these questions. And so if together we can go and propose a study like that, there used to be, yeah, well, sure there was a company, Allscripts, any of those. There was a company called Medco, which was in that business, and they got sold, but before they got sold, they had a very visionary management who was willing to run experiments for people if you went and proposed them. They were willing to run those experiments for you. And, but you had to apply each time, and I wanted to tell them, I said I could make it easier for you. We could create some kind of an API or whatever, and people could submit and run their own experiments. But he said that, you know, the reason they wanted to put their professional health services researchers in the middle of this is that it's so easy for people who aren't skilled at this to kind of design what they're looking for in a way that will come up with wrong conclusions. And so we tried to act as a little bit of a break or a little bit of advice to make sure that the studies were well conceived to answer those questions right. And I think that's probably fair enough. But we need to make some deals like that. And those are the kinds of things that, you know, together we could have the much more of an ability to be able to get impact. And doing this not just as a business, but as a data philanthropy project, right? This is something that if we could get all of corporate America to do that. Oh, and I left one thing off of my thing, my natural language question answering, remember at lunch? For those of you interested in machine translation out there in Microsoft land, I want, you know, the opportunities, when I say it takes 20 years to get to the hustings, it takes a century to get to the third world, we need to be able to translate these findings and this information to every language because it's a different planet out there. They're like 50 years behind. And in an era where we're in the internet, right? The information can flow instantly. You can ship specimens with FedEx. You can get drugs back by FedEx. There's no reason we can't deliver state-of-the-art if we get state-of-the-art information to the people who need it. So I would love to work with a group that would be like to take, you know, our content and figure out a way to internationalize it.
Great.
Okay, thank you.
Thanks a lot.
Thanks.